I'm Beth Pitel, and I'm a development technologist at the Mayo Clinic in the Genomics Laboratory. I'm honored to present on oncology knowledge bases and their utility for clinical diagnostics. All of us in the cancer genomics realm understand this problem, and we are all in this together. Given the rapid advances in technology and genomics, there are more data available in a typical diagnostic clinical assay than can be meaningfully incorporated into a clinical report. Many of us use automated pipelines to sift through results in hopes of focusing on the most clinically meaningful findings. But even with the most sophisticated bioinformatic pipelines, a significant manual effort is required to interpret genomic findings for a particular tumor. Both automated and manual data management are heavily dependent on accurate and comprehensive data and knowledge bases. In other words, we need comprehensive and accurate tools. I'll spend a few minutes orienting you all to a number of helpful resources that we have encountered in our work. I'll only have time to introduce these resources briefly, but a comprehensive user guide with specific orientation to most of these resources can be found on a new website for variant interpretation for Cancer Consortium, cancervariants.org. I will try to give you an orientation to the types of data that each resource contains, some of the more useful or unique features, as well as encourage each of you to use and to contribute to these shared resources so that we may all advance our practice and benefit from collaborative effort. I find it useful when thinking about genomic resources to categorize content as either data or knowledge. Although data and knowledge are dependent on each other, it is important to understand that data portals contain observations, like those typically seen in a results section of an article, usually without inference. These resources are essential to facilitate the generation of new knowledge. However, for example, the frequency of a mutation in a specific type of cancer does not speak to its clinical relevance and cannot substitute for what is known about a gene's function, response to therapy, etc. Knowledge bases, on the other hand, contain critically processed data, contextualized for significance and meaning, much like what you might find in a conclusion section of an article, and are often more appropriate for immediate use in clinical laboratory practice. Many of the resources I will describe have aspects of both data portals and knowledge bases. There are more features to these resources than data and knowledge, and I've attempted to code them for ease of recognition. Genomic resources may house gene-level data, chromosome-level data, clinical diagnostic, prognostic, or predictive information. Genomic resources may also contain great visualization tools. They may be manually curated by experts in our field. We've been over data portals and knowledge bases. Furthermore, many resources share their data in the form of data downloads or shared API, Application Programming Interface. API allows other resources to pull information from the resource by providing the API code, or for any programmers out there, RESTful endpoints. So the first resource I'm going to go over is CIVIC, or Clinical Interpretations of Variants in Cancer. It's hosted out of the McDonald Genome Institute at WashU, and the main data source is evidence items, or actual pieces of primary literature. They are submitted by the crowdsourced public and manually accepted and edited by the expert staff at the McDonald Genome Institute. They have many key collaborators, which help them to populate their data and allow crosstalk between CIVIC and these other resources. So here's an example of what you might see if you query CIVIC for a BAP1 mutation. So the evidence for the mutation shows up down here. So each of these rows is an actual piece of primary literature, and it's been summarized in this table. And then they assign an evidence level and a trust rating to each publication, and the criteria for these two categories is explained on the website. So I've highlighted this third row, and based on the icons here, I can tell that this paper supports resistance to some sort of therapy. These therapies in this cell right here. And if you actually highlight this, like I did in this example, and you can see that it's shaded light blue, you see a summary that shows on the bottom, which will summarize the actual paper that this comes from. And from here, you can also link to PubMed. Precision Medicine Knowledge Base, or PMKB, is hosted out of Weill Cornell Medical College in New York, has several notable features, including the highlight of pertinent negatives on their website, which is something you don't really see a whole lot with knowledge bases. It's really easy to search and browse, and they have some really well-written, curated interpretations of variants. Another feature that's available on PMKB is this Watson-esque query system called the PMKB bot, where you can type in a gene name or indication, and in this browser window here, you'll have several interpretations pop up that match your query. COSMIC, or Catalog of Somatic Mutations in Cancer, is hosted out of the Wellcome Trust Sanger Institute in the UK. It has a lot of features on this site, and it's so comprehensive. I can't possibly go into every single feature on this site, but of note, they have some really cool features I've listed here, including the 3D protein visualization that you see in the upper right, a cancer browser, a widely used gene list called the Cancer Gene Census that is complemented by the hallmarks of cancer, which is really, really cool, and you should check it out. In addition, on the horizon for COSMIC are the addition of epigenetic biomarkers and also the addition of impact calculations for all of the documented mutations on their website. Cancer Genome Interpreter is a query-based knowledge base out of IRB Barcelona. And by query-based, I mean that you enter in the mutations that you see in a genomic profile, and you submit those mutations, and you get a PDFable report that as an output. And this report shows predicted driver and passenger mutations, which is really cool, and it links to references, and it's really useful for finding treatment potential. Pecan Data Portal is from St. Jude's Research Hospital in Memphis, has several notable features, including Protein Paint, which is an interactive visualization tool. And in Protein Paint, expression data is coupled with mutation data on the same screen, which is really cool. In addition to Protein Paint, they have a really useful cohort summary. So this is the output when we query NTRK3 in Protein Paint. So here is our gene. Each of these lollipops represents a mutation, and I've selected this fusion mutation with ETV6. If you select a fusion, another really great feature of Protein Paint is it highlights your pertinent fusion, so we know which parts of the gene are involved in the pertinent fusion. In addition, you see when I highlight this fusion marker, it identifies the data points for our expression data on the right. Pecan Data Portal also has several tools in beta as of October 2017, including PecanPy, which is the pathogenicity information exchange. It's a tool for variant annotation and pathogenicity assessment. Warden, which is an RNA-seq differential gene expression analysis, so it's useful for comparing effects of knockout and knockdown experiments. There's a neoepitope prediction of immunogenic peptides in specific tumor types. There's Rapid RNA-seq for fusion detection, and ChIP-seq. And this tool calls peaks from ChIP-seq data using similar protocol to the ENCODE project. And so for more information, please visit the Pecan Data Portal website. The Middleman database is a really valuable database, especially for conventional cytogeneticists. It's hosted out of the National Cancer Institute and supported by a couple different Swedish cancer foundations. It's a monumental effort to curate this knowledge base quarterly, and 3 scientists do this on a quarterly basis. As I mentioned before, this database houses chromosome level data, which is really important for those of us looking at whole genome copy number data and chromosomes, conventional chromosomes. And each piece of data is connected to specific samples in the primary literature. So for example, if we use their recurrent abnormalities searcher for a specific indication, in this instance osteosarcoma, you'll see 2 different case numbers along with their karyotypes and links to the primary literature. Integrated Oncogenomics, or Intogen, is hosted out of IRB Barcelona, the same group that gives us the Cancer Genome Interpreter. And it uses OncoDrive tools to statistically predict driver mutations. These tools are really neat, so if you're into statistics, you should look into these. In addition, this group has developed some software called OncoPad, which is made specifically for designing cost-effective, quality, next-generation sequencing panels. So if you're looking at designing next-generation sequencing panels, maybe you want to look into this website down here. Database of Curated Mutations is out of the McDonald Genome Institute at WashU, so the same group that gave us Civic also provides this website, Database of Curated Mutations. This is also crowdsourced, much like Civic, and it's a really user-friendly interface to search, submit variants, and link to primary literature. The ICGC Data Portal provides a detailed analysis of many cancer datasets in large studies. And this is a great website for visualizing different cohorts and looking at different aspects of what's going on in these cancer genomes. So for instance, I selected a couple different primary sites, and you can see the donor distribution for each of those primary sites, the top 20 mutations in the studies that encompass these primary sites, as well as the mutation frequency in each of these cohorts. In addition, you can generate survival curves when the data is available on ICGC Data Portal, and you can investigate co-mutations using their OncoGrid function. CBio Portal is hosted out of the Memorial Sloan Kettering Cancer Center, and it holds data from large studies and cohorts, and all of these are available for download on their site. And many of these studies also have copy number data, which are really hard to come by sometimes. And you can query your results and save those results, or you can automate queries using their API. Another really cool function is you can upload your own data and use this tool, this OncoGrid tool, to look at co-mutations in your own research cohorts. The Jackson Laboratory Clinical Knowledge Base is another really useful resource. You can register for access to the free site using this link right here, and on this site, they provide diagnostic, prognostic, and predictive information at the variant level, and they also provide a clinical trial search. They also have great educational resources available on their website. So I urge you, in addition to the tutorial I provide on cancervariants.org, to check out the resources on their website. So if you query prostate cancer in this knowledge base, you'll come up with a whole host of evidence and clinical trials that are available for this indication. And on this table, you'll see molecular profile, which is also a searchable item on their website, therapies, which you can search, and different efficacy evidence here. So you can look into any of these resources. There are links to PubMed ID, and so links here and links to references on this panel as well. OncoKB is hosted out of Memorial Sloan Kettering Cancer Center, the same group that gives us CBioPortal. OncoKB is great in that it categorizes actionability of variants into 4 levels of evidence, and mutated domains within genes are identified with a query as well. So if you query P10, you can see the mutations that affect these 2 domains in P10. The dual specificity phosphatase catalytic domain and the C2 domain of the tumor suppressor protein. And then this slide is directly from OncoKB, and it explains their levels of evidence from FDA recognized and approved drugs for specific indications as level 1, down to variants that have compelling biological evidence as level 4. And in addition, OncoKB will also highlight variants that are predictive of resistance to FDA-approved drugs for specific indications. The Compendium of Cancer Genomic Aberrations, or CCGA, is hosted by the Cancer Genomics Consortium, also known as CGC, and is a collaborative effort to highlight copy number changes and clinically significant mutations that are contextualized by tumor type. This site is in need of collaborators and curators, and it is in a wiki format, so it's very editable. And all you need to do is contact somebody in the CGC and they can get you hooked up to start supplying some information for this knowledge base. This is one example of a graph that is being generated by this group to highlight copy number trends in specific tumor types. The goal is to link copy number trends and other known genetic events in cancer to pertinent knowledge pertaining to diagnosis, prognosis, or therapy. On this slide, I have compiled all of the attribute icons for each genomic resource that I've covered on previous slides. I've done this to identify trends and to help direct towards the appropriate resources to better answer your genomic inquiries. The first thing I notice when looking at these data is the fact that all of these resources contain some degree of gene-level data, but only a couple contain chromosome-level data, which is useful for those of us who interpret conventional chromosome studies. Another resource that is highly used that I did not cover as a part of this project is the Atlas of Genetics and Cytogenetics in Oncology and Hematology. This is a well-trusted resource with a wealth of genomic information that is particularly helpful for the conventional cytogeneticist. You can access the Atlas at atlasgenetics.oncology.org. Additionally, in order to use data portals and knowledge bases effectively, you need to know what information to expect when you access these sites. It might be important for you to know which of these sites contain critically processed interpretations of the data versus sites that offer actual data that you can in turn use for your own analysis. Look for the brain icon for sites that offer knowledge, which we are defining as critically processed interpretations of the data, and look for the purple squares indicating data portals. You may need to extract data for your own research and development. If so, it's important to know which of these sites offer data downloads indicated by the aqua arrow or shared API indicated by the green circle. So while these resources are powerful and helpful, it's important to recognize that there is very little overlap in their data content. This table is from a recent publication in Nature Genetics from the group at the McDonald Genome Institute at WashU. As you can see, these databases with similar end goals only share a fraction of the literature with the other data sources. It illustrates the problem of a lack of a comprehensive resource as a result of A, an enormous amount of existing literature, and B, the huge amounts of effort it takes to sort through the literature and deposit meaningful findings into a knowledge base. In the next 2 slides, the colored circles represent genomic resources for which I've created user guides as part of this project. The gray circles represent genomic resources that I was not able to cover. And the lines between resources represent connections between the resource in the form of shared data. There is interconnectivity between some of the resources, but it is currently difficult to know if you have comprehensively evaluated the available knowledge regarding a particular tumor or mutation. Today, this interrogation of current knowledge requires multiple inquiries. Of course, the ideal future state is one where knowledge is shared across multiple available resources and summarized in a way that's desirable for the end user, with accurate and automated queries. My pie in the sky thought is that the comprehensive resource might be user definable and collaborate with many resources. So let's work together to make this happen. So integration of resources can happen in a few ways: shared API, downloadable data from the resource, and inter-resource collaborations. I'm hoping this presentation and the tutorials I provided will facilitate more exposure to these resources, stimulate collaboration and data and knowledge sharing, so that we can all advance each other's practice. For more information on collaborative knowledge-basing efforts, visit these two websites. VIC, or Variant Interpretations for Cancer Consortium, has the support of several knowledge-base leaders, and this group is attempting to standardize annotation across knowledge bases and to make it easier to query all knowledge bases involved in this collaboration. CCGA, or the Compendium of Cancer Genome Aberrations, is also a really great collaboration. And if you're interested in getting involved, please contact a member of the CGC or the Cancer Genomics Consortium, or visit cancergenomics.org. So there are genomic resource guides available, and these are available currently on cancervariants.org. And I have some PDF tutorials for several of the resources I went over in this presentation. So please use these genomic resources. You can support these resources by accessing their websites, providing feedback on the websites, and contributing to their efforts. And there are more details focusing specifically on how you can make contributions to these resources on the PDF user guides. I have a lot of people to thank for the success of this project, including our GOATS team at Mayo Clinic, or Genomics of Oncology Annotation team, all of the knowledge base experts who so graciously offered their time and insight for the success of the tutorials, the Cancer Genomics Consortium, or CGC, for allowing me to speak at the 2017 annual meeting and to make this presentation, and to the Variant Interpretation for Cancer Consortium, or VIC, for making the knowledge-based tutorials publicly available on their website. And if you have any questions pertaining to these knowledge bases, please feel free to direct them to the specific resources in question. So see the tutorials for the contact information, and if they're not available, go to the websites and seek it out, or you can email me for contacts. If you have any suggestions or comments about this presentation, I'd be happy to take those as well, and you can email me directly. Thank you so much.